form, and he's going to tell you about that now. Hagan, the floor is yours.
Well, welcome. I was told to make this a little bit autobiographical and include some science, so I've done my best to do that. All right, so I was born in a place called North Wales. You're in England now, but if you go left a bit, you'll end up in Wales. And I was born in the north of Wales in a little town called Prestatyn, and this postcard was made before I was born, but I lived in this house here, and I stayed in Wales during my youth. There's the house, and there's my brother and his me. My brother decided to get rich, so he became a poet. I became a scientist. And this North Wales, Prestatyn, North Wales, is a very out-of-the-way place, but we did have in 1962 the Beatles, before they were famous, because Liverpool is quite close by, visited our town and gave a concert, and that was the end of 1962. In 1963, they had their hit record, Please Please Me, released in January, and they never came back to Wales. And indeed, after a while, I took the train away from Wales and came to Oxford, and I'll tell you the rest of the story as we go along. So when I came to Oxford, I came to Balliol, which you can see here. That's where we are now, of course. And I studied organic chemistry and spent a lot of time in this lab on South Potts Road, the Dyson Perrins laboratory. But in Oxford, we have a fourth year of chemistry, which is a practical year, and often that became the first year of a person's PhD. And my supervisor at the time told us halfway through the year that he was moving to Harvard. So the entire research group moved to Harvard, and I actually did go to the States on this boat, and I ended up doing a PhD at Harvard. It took about twice as long as it would take to do a PhD here. Here's my boss, Jeremy Knowles. And then after that, I did a postdoc at MIT. I was too lazy to move house, so I just went to MIT, which is down the road, and did a postdoc with Govind Khurana at MIT, who's famous for the genetic code, but at that time he changed to start working with membrane proteins. As Dennis said, that's what I'm interested in, and that's how I got into that field. So, to cut a life story short, I did my postdoc, then I ended up as an assistant professor at Columbia University. Then I moved to a small research institute, the Worcester Foundation in Massachusetts. And finally I moved to Texas A&M University. And then in 2003, when they built this fantastic new lab on South Potts Road, I came back to Oxford. So I'm indeed interested in membranes, and so there are lots of membranes in cells. There's the membranes that surround cells and all the organelles within them. And I'm particularly interested in the proteins that sit in these membranes. So these membranes are formed of lipid bilayers, of course, but they wouldn't be able to do very much without proteins in them. And so there are all manner of proteins in the lipid bilayers, and they can transmit signals, they can act as enzymes, they can move things back and forth across the membrane, and so on. But the particular class of proteins that I became interested in are called protein pores, and they're often secreted as toxins by bacteria, or they're found in the outer membrane of gram-negative bacteria. And there also our immune system has encodes some protein pores as well. So here you can see a typical one. This is called streptolysin O. This is a little lipid vesicle. And you can see this protein is forming a big pore in the membrane. And these pores are actually quite big. They're several nanometers across. So we like to call them nanopores. And I hope to convince you that there's some interesting basic science behind these nanopores. I actually won't talk about that very much, but they can also be very valuable in biotechnology. So just to tell you a little bit about our lab, so we're about 24 people, graduate students and postdocs in the lab, and also one lab manager who's sitting at the back, so she can answer any difficult questions if you have them. And basically, ours is a so-called interdisciplinary or multidisciplinary lab. So we're in a chemistry department, but we like to throw any type of science that we can understand at solving the problems that we're interested in. So we have some people who are really mainly chemists, some who are biologists, some who are physicists, all working together on various different basic science problems in the lab. And one problem that really interested us was how these protein pores get into membranes to begin with. So, for instance, the pore-forming proteins that are secreted by bacteria are secreted into the medium. They're perfectly water-soluble. And it turns out that they bind to the membrane, then assemble, in the case of the protein I'm going to talk about, into a heptamer, a pore that has 7 subunits, and then they eventually pierce the membrane. But it was working on this very basic science problem that really got us thinking about how we could use these pores in biotechnology. So I'm pretty convinced that a lot of the advances in applied science come through doing basic science. I won't tell you about our basic science today, but I'll tell you about 2 examples of applications. One is using these pores to do ultra-rapid DNA sequencing, and that came from the company Nanopore. And also more recently, I'll tell you very briefly at the end, we've been making 3D printed synthetic tissues which require these protein pores to function. That resulted in another company called OxyBio. So here's one of the proteins that we're interested in. It looks like a very delicious mushroom. It's called alpha-hemolysin. It's secreted by gram-positive bacterium, Staph aureus. It's a very simple protein. It has just 293 amino acids. And this protein will assemble spontaneously to form this heptamer that will go through the lipid bilayer. So the lipid bilayer would be here, and I've just shown each subunit in a different color. And this pore, as I mentioned earlier, has nanometer dimensions. So we can start off with a big needle, and if we get smaller and smaller and smaller, we'll end up with a protein pore like the hemolysin pore, and that has about 1/50,000th of the diameter of a human hair. And that diameter is comparable with the kinds of molecules that we're interested in, like DNA that we'll be analyzing. So the DNA will just about fit into this pore. If this pore was any bigger, it wouldn't be useful for analyzing DNA sequences, and if it was any smaller, the DNA wouldn't be able to get into it. Another important thing is that we can measure currents. These currents aren't carried by electrons, they're carried by ions, like potassium and sodium ions, that pass through single copies of these pores. So it's possible to make an apparatus and have a little hole in a piece of plastic that separates 2 chambers and get one of our pores to go into that lipid bilayer, and then with electrodes on each side we can record the current that goes through a single copy of the pore. And that was really the basis of the technology that's used by Nanopore. So when we first set up Nanopore, we were interested actually in analyzing small molecules at the single molecule level. And we could use protein engineering, for example, to put a cyclodextrin host molecule inside the pore, and that host molecule, once it's in there, will bind small organic molecules. I've just shown ibuprofen here. And when that molecule binds, it will change the current that goes through the pore. And amazingly enough, in this case, you get a different signal, amplitude of signal, for the different optical isomers, the right or the left-handed forms of this molecule. And that's because the binding site in here provides a chiral environment. So this traces 50 milliseconds across here, 1/20th of a second, and you could use that to, in this case, determine that you have a 50-50 mixture of these two optical isomers in your solution. So the company was founded around that in 2005, Obser Nanopore, and I would just say in passing really that Oxford University is a very good environment for academics to get involved in entrepreneurship. So we have a technology licensing office, Oxford University Innovation, and we have various companies and university institutions that can provide initial funding for companies, and we have good science parks and a lot of strong supporters and potential employees, and that really led to the, at least in part, to the success of this company. So at the beginning of the company, there were really three Oxford people involved, me and two colleagues, Gordon Sangera, who's still the CEO, and Spike Wilcox, that still works for the company. And you can see that 5 years on, people were still pretty skeptical about my role in forming this company. So it says, Bayley speaks in the laconic baritone of a late-night radio DJ. If he has an entrepreneurial streak, it's not obvious. So I guess I can make two comments about that. Oxford Nanopore now has over 250 people working for it, and it's raised 350 million pounds in funding, and spent most of that, I should say, too. And then, of course, if I didn't have a day job working at Oxford, I'd really like to be a DJ, so maybe work on that. Maybe Sonny can get me that kind of job. All right, so the company was set up to detect small molecules, but a year and a bit in, we decided to pursue this goal of sequencing DNA. So the idea is very similar to detecting small molecules, but instead of one molecule binding at a time, a single strand of DNA is passed through the pore. It actually requires an enzyme on top of the pore to do that. And the idea is that you'd have a little reading head inside the pore, a little binding site, and read off each base at a time as it comes through. Of course, in reality, it's a bit more complicated than that. But after working on this for a long time, over 7 years, Oxford Nanopore were able to produce a DNA sequencer that works on that basic principle. So this was really announced first in 2012, and it really did take a long time and a huge effort. And one thing you'll notice is the laptop here, that the sequencer is very small. So this machine, we can call it a machine, is about, well, I used to say it was about the size of a cell phone, but I've noticed that cell phones are now getting bigger and bigger. So it's now smaller than a cell phone. And it's a parallel device. If you just had one of these pores, you couldn't sequence something like the human genome very quickly. It's got 6 billion bases in it. So right now, the present model will run about 500 pores in parallel. It takes about 5 milliseconds per base per pore, and you get the data in real time. It will do very long sequencing reads. So for people who don't know about DNA sequencing, most sequences nowadays just sequence a few hundred bases each time a piece of DNA is analyzed. But this will analyze very long pieces of DNA, and it's very easy to operate, and it's also a portable device, and that's very important, I think. And just to accentuate that, recently this device, which is called a MinION, has been used to sequence viruses actually in the field. So it was taken to West Africa, to Guinea, to sequence Ebola virus, and there was a paper on that in Nature last year. And then more recently it was taken to South America to sequence Zika virus, and just a couple of months ago a paper on that was published. And the importance of that is that neither of these viruses are single entities. So you can see from the Ebola virus here, there's an amazing family tree of these viruses, some of which are more dangerous than others. There are literally hundreds of different forms of this virus. All right, so let me finish about telling you about our second area of work. So I don't spend much time with Oxford Nanopore now. We have a new company called OxyBio that's involved in making synthetic tissues. And this is how they look. But the basic idea behind that, just like Oxford Nanopore, is a very simple idea. So if you take an oil, this could be a silicon oil or hexadecane or something like that, and have a lipid molecule in it. These are the same lipid molecules that you would find in the membranes of cells. And you put a tiny water droplet into that oil, a monolayer of this lipid will form around the droplet, with the head groups of the lipid inside and the tails outside. And if you bring two of these together, a lipid bilayer, the same kind of lipid bilayer that you'll have on the surface of a cell, will form between these two droplets. And originally we used this to miniaturize these single channel recordings that I told you about earlier. The apparatus I showed you earlier will have about 1 ml of liquid on each side. These droplets here are a couple of hundred nanometers, so it reduces the volume that you need by about a factor of 10 to the 4. So you can use very expensive reagents or very rare proteins in your experiments using this technique. But we realized at the time that just as if you could join two of these together, why couldn't you join 3 or 4 of them together, or 100 or 1,000 or 1 million of these together, and make interesting materials from them. So what I'll show you how we do it in a minute, but what we've ended up doing is making these little building blocks of material, and you can just about see each little droplet here. In this case, these will be picoliters in volume, about 50 picoliters, and each of these droplets is joined to its neighbors through a single lipid bilayer, and we can functionalize those bilayers by putting our favorite protein or other membrane proteins into the bilayer. So this is done by 3D printing, and I actually got the idea for this by occasionally take some time off work, and was reading a magazine, The Economist magazine, which is a very good magazine, and there was an article on 3D printing where someone had printed a violin. I don't think that works very well at this point. And you can print sort of gloves and all sorts of things. And I thought, why don't we make a 3D printer? Because no commercial 3D printer would be able to print these droplets to make these things. And this was done by a very talented student called Gabriel Villar in the lab. I just sit behind my desk all day filling out forms and doing things, so I couldn't possibly have done this myself. But we had a very talented physics student who actually built the hardware and the software to make a 3D printer to print these tiny droplets. And again, without going into details, these are the kind of things that you might get out. So here's an object, if you have 1 millimeter across, that looks different depending whether you look in the X, Y, or Z direction. You can see these different views here. And then here, on purpose, something printed in 20 layers that's meant to look like an arborizing neuron, for example. This is how it comes out once you've printed it. So you can functionalize these materials. So one example is we can put our pore-forming protein into the green droplets. They've just got a green dye in them. And that will mean that these droplets are connected to neighbors through bilayers that contain these protein pores. So they will carry a current through them if you put electrodes onto this material and apply a potential. And this current will flow directionally and very quickly, and that's exactly what a neuron does. It's a completely different mechanism to a neuron. Dennis is probably horrified here, but it's fast and it's directional, so it's a way of getting a signal from one side of this entity to the other extremely quickly. Another thing we can do, it's also at a very early stage, but we can get these entities to fold up, and that the way we did that at the beginning was to print droplets. The orange ones have high salt, the blue ones have low salt, and once they're printed, water just by osmosis flows from the blue ones to the orange ones, and then these entities can fold up. And you can get this to happen in 3 dimensions. So here we printed something like the petals of a flower, and they will fold up into a sphere. And if I'm lucky, actually, no idea how to get the cursor on this, never mind. Oh yeah, here we go. This is actually a computational model of how that should happen. And then you can see it happening in real life here. So, and right now we're trying to make this happen faster and reversibly, so we could make materials that would resemble muscle. And based on that technology, we founded a second company just in 2014, and the overall idea is that we can print synthetic tissues like I've just shown you, and couple these with living tissues and be able to control them from the outside world, either optically or by using inserted electronics and so on, and eventually perhaps use these to repair or enhance organ function. So that's my little story. Thanks for listening.